Good afternoon. My task this afternoon is to talk about the why of disinfection. Why is disinfection such an important issue for you as potato growers? And of course, when we talk about the why of disinfection, we're talking about potato diseases. And we understand and we know, and you probably know it's better than anyone else, that potatoes are particularly a challenging crop in regard to diseases, because potatoes are grown from vegetative part of the plant rather than the true seed. Crops you grow from true seed, you exclude a lot of diseases, but by propagating potatoes from part of the vegetative growth, you're carrying along a lot of diseases, both internally and externally in potatoes. Furthermore, the seed tubers that you grow are produced in soil, and many of the pathogen propagules where the diseases start from are born in the soil environment. And there are a lot of diseases you need to deal with. Over 100 diseases have been described for potatoes. I've seen numbers as high as 300 different diseases of potato. These diseases are caused by different types of organisms. There's bacterial diseases, fungal diseases, viral diseases, and diseases caused by nematodes. And it's important to understand that the nature of the diseases is determined by the type of organisms that cause the diseases. Today we're talking most about bacterial diseases, but not exclusively. It's also important to understand that the inoculum and the dispersal of the disease organisms are different from one another. Some pathogens are airborne. You think of late blight and the spores blow around in the air, or you think of viruses that are spread by aphids that fly around in the air. Other diseases are more soil-borne. For example, many of your fungal diseases have spores that survive for a long time in the soil environment. Other diseases are waterborne. For example, the soft rot organism survives very well in water, can come down in rainwater or through irrigation water if surface water is being used. Other diseases are tuber-borne, where they're carried on or inside the tuber. And here, of course, we think of bacterial ring rot as an important disease that is tuber-borne. I've also introduced a new category here, multi-borne, because many diseases aren't just tuber-borne or just airborne, but they can be soil-borne and water-borne, or soil-borne and tuber-borne, or perhaps in all the different categories. But as you control these diseases, it's important to understand where is the inoculum, where is that disease coming from, and where does it persist. I also want to draw your attention to the different cultural practices that you implement for controlling potato diseases. Some of these involve a disinfection step, others do not. Cultural practices, for example, when you use your equipment for planting and harvesting, disinfection is an important component of the cultural practices that you use. When you use pesticides, it's less so. Pesticides is use of fungicides for controlling, again, late blight, or you use insecticides to control your aphids, to control virus diseases. Quarantine, we often think of quarantine in terms of preventing organisms from entering the country, but you can also think in terms of quarantine of your own particular operation, your own farm, preventing an organism from getting in the first place. Clean seed, maintaining clean seed, of course, disinfection plays an important role in the process of producing and maintaining clean seed. And of course, there's also resistant cultivars. But as you know very well, many of our diseases don't have good resistance. There are not good resistant varieties available for many of the diseases that affect potatoes. Now I want to switch gears a little bit and talk about bacterial ring rot. And I know some of you probably have never seen ring rot because we have essentially eradicated it from the island industry. We don't see ring rot around. But it's a particularly devastating disease if it gets out of hand because it can destroy the entire crop. And the only way to control bacterial ring rot is through a control program of avoiding getting it in the first place and keeping it under control if it does exist, because Ring rot is a particularly difficult disease to deal with. The symptoms of the disease are not always clear. In fact, symptoms may be absent altogether. You can have the disease in your operation for a year or more and spread it around without knowing that you are a carrier of the ring rot disease. But there are typical symptoms. There are very typical symptoms as depicted in these couple pictures here in the foliage of potatoes where you have the leaf symptoms. Certain varieties will also show this rosetting or stunting where the internodes are very short. Something perhaps we don't think about often is the ooze that can be expressed from infected stems. And this is part of the danger with the ring rot disease because that ooze that can spread on your equipment contains thousands, even millions of bacterial cells. Same thing is true for the tubers. You can have very typical ring rot disease. Of course, that's why it's called the ring rot disease. The disease affects the vascular tissue and the ring of the tubers. However, tuber symptoms are not always typical. Secondary organisms get in and can greatly modify that typical kind of ring rot symptoms. And again, I draw your attention to the slime that can be expressed from an infected tuber. And again, this slime can contain many, many bacterial cells, and this is what gets spread on your equipment, and this is what you need to address in your cleaning and disinfection protocols. This is a ring rot bacterium. It's called Clavibacter michiganensis subspecies Epidonicus. Name is not important, but it is important for you to get a mental picture of what the ring rot bacteria looks like. It's a typical bacteria. It's really a short cylinder, and each of these cylinders is a ring rot bacterium. You can have great numbers of them. This is under the electron microscope. This is another picture under the electron microscope showing the interior of a ring rot infected potato stem. And you can see some of these vessels here in which the nutrients flow from the roots to the leaves. And in this vessel here, it's just chock full of bacterial cells. Huge numbers of bacterial cells are present inside a ring rot infected plant. Also keep in mind that these bacteria are very, very small. You need 25,000 of them lined end to end to cover 1 inch. So you can imagine how many bacteria would be present within a square inch of that slime that we saw in the tuber and in the stem. Also, bacteria can grow quickly, relatively quickly. The ring rot bacterium, as far as bacteria grow, does not grow particularly fast. But in 24 hours, a single cell under favorable conditions can increase to about 64 cells. However, in a week, a single cell can grow to over 4 trillion cells. You get huge, enormous populations of these bacteria. And that's not all. It used to be we thought of the bacteria as being single individual cells. But now we know that bacteria grow as biofilms, this amorphous type of substance in which bacteria are embedded in the slime, the polysaccharide matrix that the bacteria produce. And they're protected within that slime. The ring rot bacteria on their own are a very stable type of organism. They have a very thick cell wall that is very resistant to drying, and these bacteria can survive for not just weeks and months, but for years in dried slime on equipment, in cold areas. If they're kept cool and dry, these bacteria can survive for a long, long time, even more so when they're embedded in the biofilm matrix. Furthermore, the infection process that technically are very few bacterial cells can start a single infection. A single bacterial cell is all that's needed to start a ring rot infection. And when these living bacteria come in contact with potato tissue, they will start to grow. Therefore, it is of utmost importance that no ring rot bacteria be of any surfaces with which potatoes come in contact. And that's why disinfection is such an important practice on your farm. So What to do about bacterial ring rot? In the first place, ensure that ring rot never gets on your farm. Do everything possible that it never gets on your farm. If it inadvertently were to get on your farm, you need to have a clean operation to make sure that it gets taken care of at the very early stages before it starts an infection. How do you prevent ring rot from getting onto your farm? And the big thing is buying clean seed. Where does your seed come from? Know your seed supplier. Know that he runs a clean operation, because in so many instances when ring rot has been found on the island, it's been traced back to the seed source. However, it could also get in on dirty equipment. If you get used equipment on your farm, if you share equipment with neighboring farms, it needs to be disinfected before it comes on your farm to make sure you don't bring ring rot in. Same thing with trucks that come into your farm. Be sure they are clean before you bring them onto your farm. Also think of people that come onto your farm, come in contact with your crop, whether it's a buyer or a neighbor or a cousin or whatever. People can also be carriers of ring rot if they have been exposed to ring rot on another farm, carrying it on their boots or their cutting knife or gloves or whatever you might have. So what to do about inadvertent ring rot? Run a clean operation. Call the biosecurity program, if you will. Dave is going to talk about the biosecurity program, which really is all about running a clean farm, clean and disinfect all equipment and storages on a regular basis, and dispose of all washings and stray potatoes safely. Now, the benefits of running a clean operation, of cleaning and disinfecting, extends beyond just bacterial ring rot. There are other diseases also that benefit, or the control that benefits from running a clean operation. And I just want to say a little bit about these other diseases, the first of which is potato blackleg. Things used to be simpler than they are now. There was a time when all the blackleg in the Canadian potato industry was caused by a single bacterium, Pectobacterium antisepticum. It used to be called Erwinia antiseptica. It is still the main culprit for blackleg, but there are other species around now that also cause blackleg. We've isolated them. We've seen them here in Canada. Pectobacterium brasiliensis. It's called brasiliensis because it was first described from blackleg infected plants in Brazil. There's also Pectobacterium. We used to call it wasabi because it was first described on horseradish in Japan. Now, with the new molecular methods, we can differentiate the strains from horseradish from those on potato. We're calling them Kelmani after Arthur Kelman, who was my major professor in Wisconsin when I did my studies many years ago. I also list here the Dickia dianthicola and Dickia solani, not because they're a problem here in Canada, but they cause blackleg in Europe. And they increasingly have caused blackleg in the European industry. They've spread it all around. It was just Dickia dianthicola. Now they have another species also causing blackleg in potatoes in Europe called Dickia solani. This particular strain probably came from the flower bulb industry in the Netherlands, because this species also is associated with hyacinth bulbs in Europe. For the sake of completeness, I've also included Pectobacterium kerativorum, which causes the aerial stem rot of potatoes. And aerial stem rot is distinguished from blackleg in that blackleg always starts from the seed piece. It's characteristic of the disease that it always originates from the seed piece. Doesn't matter which of these species causes the disease, it always comes from the seed piece. Stem rot does not. Aerial stem rot starts from wounds in the stem caused by cultural practices, insects, fungal infections, and so forth. This is just a picture of the typical Antiseptica blackleg. Tend to call this early blackleg, where early on in the season your plants are short and stunted. If you pull them up, you see the lower part of the stem is decayed. Later in the season, blackleg occurs. Of course, you get the very typical blackleg stem. We can test the strains quite easily in the laboratory conditions. Wasabia blackleg, we have seen that. Often this species occurs in conjunction with the Atroseptica, but we have seen it by itself in Canada as well, causing the very typical blackleg symptoms that we see with the Atroseptica blackleg. So there are other species now in our industry that is causing this disease. This is Dickia blackleg. It looks a little bit differently here, and that goes right up the inner part of the stem, the inner pith region, and this tends to occur with the Dickias at the early stage of the disease. But as the disease progresses, it'll burst out to the outside and looks just like blackleg. It's indistinguishable from blackleg. I've been in, over the years, in many fields in the Netherlands where they have both the Atroseptica blackleg and the Dickia blackleg, and you just can distinguish the two on the basis of symptoms, although this tends to be very typical of the Dickia type blackleg. Here again, this is aerial stem rot. The stems don't, the diseased stem don't become as black as it does with blackleg. They're browner, and as I said earlier, they are initiated from wounds in the stem rather than coming up from the seed piece, as is so typical of the blackleg disease. But all these organisms that cause blackleg can also cause tuber rot, tuber decay. Is a very typical blackleg shown here. The blackleg symptoms can be modified readily by other organisms, secondary organisms that come in. You can have all sorts of rots and all sorts of types of symptoms. Also very peculiar about the blackleg organisms, they like the lenticels, and they can go in and under the right conditions, can be in the field, especially in storage, they can cause this lenticel rot, which can dry out in storage and become a condition known as hard rot, but that's also quite typical of the soft rot organisms. Like I said, the blackleg is a bacterial disease. Like the ring rot bacterium, it's a bacterium, it looks like a cylinder. The blackleg bacteria, they have all these flagella where it can swim around. The ring rot bacteria doesn't swim, it's non-motile. But the cell wall of the blackleg bacteria isn't as thick and sturdy as it is for the ring rot bacteria. So these bacteria will survive weeks and even months on equipment, smeared on equipment, but not for the years and years that the ring rot bacteria survive in the dried state. There's also viruses to think about. Many of the viruses in potato are also mechanically transmitted. This is the PVY virus. There's many different strains of PVY, and you see the severe types of symptoms that it may cause. This is caused by a virus, very different type of organism than a bacterium. And it's not only PVY, there's a whole list of viruses that occur on potato. Now, not all of these are mechanically transmitted in plant sap. For example, the leaf roll virus is not, and the tobacco virus are not mechanically transmitted, but the other viruses can be transmitted via plant sap. So if infected plant sap is smeared on your equipment, the contact with the next batch of seed or whatever the situation might be can transmit the virus within the crop or from crop to crop. And this is not all. There's also some lesser known viruses that are not widely present in our industry, but these viruses too have been detected in Canada at some point or another. And by having a clean operation, you prevent them from building up and ever becoming a problem. Again, not each of these viruses are mechanically transmitted, but most of them are. And I just want to emphasize the fact that we don't want a buildup of additional viruses that may affect your crop. So again, a clean operation is important. Finally, I want to talk about fungal diseases. This is a picture I got off the internet of a screening of fungal spores from soil. And it emphasizes the fact that the soil contains spores of many, many fungi, some of which are potato pathogens. Some of the diseases like pink rot, fusarium, verticillium wilt, and as you know, potato wart, their spores all survive in the soil, and many others besides. These are the diseases caused by those particular organisms. However, I do want to emphasize that these diseases cannot be controlled in their entirety by a clean operation. These diseases will be around, but you can prevent the buildup and the spread of these disease pathogens by cleaning and disinfecting on a regular basis. So let's get back to saying a few things about disinfection. What does it mean? Disinfection is the killing of disease-causing microorganisms using various agents such as chemicals, heat, and radiation. I think in our scenario here with the potato industry, we're dealing really with the chemical disinfectants. Why disinfect, of course, is to kill potato disease-causing microorganisms that are carried by tubers and smeared onto surfaces or present in soil residues from where they can be spread to healthy seed tubers. 3 steps have been identified for the whole process. Step 1, the rough cleaning. Step 2, the pressure washing. And step 3, applying disinfectant. And there's no sense in doing step 3 unless step 1 and step 2 is done. Disinfectants are very quickly inactivated in the presence of organic matter and soil. So if your surface isn't clean, don't waste your time doing disinfection. These are just some slides to emphasize the point I got from the website of the Canola Council of Canada. Rough cleaning depicted here, pressure washing, and finally, and last of all, disinfection once your surfaces are clean. Cleaning and disinfection. The cleaning part involves removing of the soil and the plant debris. Okay, and this really removes 99% of the pathogen inoculum. Most of that's removed. But that 1% you can't remove by cleaning alone, and that's why the disinfection is needed. And if you're thinking, well, 1% isn't much, but 1% of 1 trillion bacterial cells is still a large number of cells. So disinfection is important to kill that last amount of inoculum that is left on your surfaces after you've cleaned and pressure washed the equipment. Again, disinfectants do not work on dirty surfaces. You need enough contact time for that disinfectant to penetrate into the biofilm, into the organisms to actually kill them. We also note that some surfaces are easier to disinfect than others. On the farm site, you have smooth surfaces, such as polycarbonates or other plastics, and steel, which are far easier to disinfect than porous surfaces that contains all these nicks and crannies where the organisms can hide and survive. Wood is particularly a difficult surface to disinfect because all the microscopic openings that there are. Killing biofilm bacteria, again, is difficult because of that slimy matrix in which they survive. And when that dries out, disinfectants don't penetrate readily, so it's important that you blast away most of that biofilm with your pressure washing and then make sure the exposure of the disinfectant is for a long enough period of time. Just a couple other points to consider. That seed cutters are particularly important to disinfect because the cutting knives and the rollers can harbor these microorganisms, the disease-causing microorganisms, and they come in contact with the fresh surfaces of the potato. So they're an ideal vehicle by which inoculum can spread from tuber to tuber. Experiments have shown with blackleg, if you cut one blackleg infected tuber with a knife, you'll spread it at least to the next 100 tubers that is cut with that knife. We also want to get in a word on foot baths. Foot baths with disinfectants are good, but when they get dirty, they are not good because the disinfectants then become inactivated and your foot baths can then serve as a means of spreading the disease pathogens around. So keep that in mind. If you have foot baths on your farm, maintain them. Maintain them so they stay clean, so that they actually do what they are supposed to do. So let me just summarize my message for today then. The problem we're addressing are the disease pathogens. Bacteria, first of all, ring rot, very important, but also for blackleg. And there's also advantages for controlling viruses and fungi to keep your operation clean. Again, steps 1 and 2 of the cleaning and disinfection process: remove all soil and plant disease and blast away the biofilm material. I had space on this slide, so I put this picture in again to remind you that this biofilm you've got to blast away with your pressure washing, because that's not going to get disinfected. Your disinfectant won't penetrate that well enough. You have to get it down to this level where there's just a few bacteria left on your surfaces, and that's what you can disinfect. And then, so apply only to clean surfaces because the dirt neutralizes the disinfectant chemicals. Use the right concentration. There's a fairly standard disinfectant being used here, so make sure your concentration is good and keep the surfaces wet long enough. And then for my final slide, I had this as a 3-step process to prevent bacterial ring rot: detection, cleanup, and prevention. Disinfection, cleanup and disinfection is so important of the cleanup process and the prevention process. Thank you.